into different parts, right? So this needs it to exclude or partition the niches in the system. And there's a whole bunch of different versions of this. All of them basically assume that there's that you're just, this is niche exclusion stuff. Okay. This is, on this part of it, so this region of your space, okay, is what we would call a neutral theory or maximum theory of maximal theory of ecology. Sorry, I'm not doing this right. Metabolic theory of ecology, right? So MTE sort of views of this. And the idea here is that this region of space is where there's a certain amount of entropy. So if you go into a system, right, there's a certain amount of free energy, let's call it, or potential free energy. And what you can do, what you assume basically in all these things is that a whole bunch of stuff comes together and it uses up all the available energy in it, okay? And that can be divided differently amongst different species, okay? But it ends up in this part of it. Can you guys see that? It doesn't matter, each cell, so that's what like the things Tracy does, it doesn't really matter what the identity of the bacteria are, right? Okay. Autotroph, heterotroph matters, right? But if we're just assuming this is all heterotrophic, it doesn't really matter what this line looks like. So you don't have, you could be right here. These could all be E. coli, and they would all be using all the LB, right? Or you could have a whole bunch of E. coli plus a whole bunch of staph, and they would try to use up as much of that region as possible, right? Does that make sense? Okay, so you end up in this region of the thing. And then this is what you would call viral information, okay? Because in this case, it matters a lot what the species are, right? So not only, so it's not just the cells, so it's not the number of cells that are mattering here. It's actually the genetic information because you're pulling things this direction. Can you guys see that, right? So basically you're becoming less efficient from this point of view. And the further you are, like, so if you're way down here, right, you have a whole bunch of stuff, but you're not doing a very good job of using the amount of energy there. Good? Do you guys see that? Yes?
Does that have anything to do with, like, the reason that viruses aren't osmotrophs and they're not taking nutrients out of the system and just kind of changing? Is that?
This doesn't matter whether you're a virus or not at this point, right? Anything can do this, right? So any cell could, so we could not even talk about viruses, right? So these guys are just breaking down physical information and turning it into the information inside a cell. So they're organizing a cell, okay? What matters here is that, in this case, they're trying to get, every time they add a cell, right, they're getting more and more, they're moving this direction. Here, they're doing a worse job of using the information, right? So if you had a whole bunch of species and you did a really bad job of eating the amount of stuff that's there, you'd end up way over here, right? So you're bringing more information into the genetic component, not into the disruption of physical information. All right? And then if you do this, right, so this is the calorimetry stuff, right? So you can actually do it. So we went through this the last time. So you guys remember this, I hope, right? So this is basically Ishmael's system. You have, in his case, a Wheatstone bridge, thermistors, and two vials that are insulated from each other. And what you're doing is you're measuring the temperature change between these two components. So in this side, you only put the water, okay? And in this side, you put the water plus the cells. And then if you measure the change in temperature, this is what you end up with, right? So that's how much work basically these cells are doing is how much physical information they broke down to make new cells, right? And then if we do it with the viruses, we end up with this, so you guys remember this one. So basically you do, and this is really becomes key, right? Because what happens here is that you ended up with, so we added food, right, to the system, which is the DOM. So we filtered out everything here. Then we add the inocula with the microbes by using the PIC45. And then on one side we add the live viruses, and on the other side we add heat-killed viruses. And not surprisingly, when you have viruses, the total number of cells go down, right? And then when you don't have them, you get more cells. Okay? But what is surprising or useful is that you actually get more information, more heat coming out of it, right? When you have the viruses there. So less cells, more heat. Good? Does everybody see that? All right, and so that's how that's calculated.
You weren't comparing the heat with microbes only and microbes and viruses.
What was that?
I didn't think that you're not showing the heat output of the microbes only.
Yeah, that's exactly what we did. So we did here, we did it with just the cells versus cells, right? But then we switched the experiment, and all we did was cells and cells plus viruses.
Are you showing the difference? Because
Right, so then this is the number of cells that we end up with, right? And then this was the difference in heat that we measured between those two things. So that's exactly what we're measuring. Right? So that's the heat, extra heat released by the viral fraction. Good?
I thought that one was showing that when you had less to begin with, you got more, but not that you were comparing
Now we were actually comparing this. This is one experiment right here. Right? Does that make sense? You see? So we were actually comparing directly to see do we get more or less heat production by having the viruses there. So you get less cells, but you get more heat production. Good? Okay. And then if you just extrapolate this, right, this is actually, this is the numbers that we measured. It's probably a little artifactual because we did all of this. Remember, there's the bottle effect, so you get more rates, things start going faster. But if you actually look at that, this is actually a ton of energy. It's a lot, right? It's being released by the system. And it's literally in this range, right? It's all of the fossil fuels versus just what the phage are doing right here. And if you were to, even if you were to say these rates are off by an order of magnitude, they're still really large numbers. Yes, sir?
So viruses increase the heat of the system, correct?
Yeah.
So then could you use that to, like, let's say you find a planet out in space and you have a predicted heat output of that planet. Could you use this kind of method to look for life?
No.
No.
No, because it's so small.
It's so small.
And the amount of heat hitting the surface, right? So it's,
But isn't the amount of heat could be coming from the number of generation cycles you went through versus just information?
it is the number of generations, which is still information, right? It's just destruction. What you're measuring is destruction of physical information to give you biological information, right? So its DOM is going away to give you viruses and cells. Right? So the number of times you do that is why you get more heat here. Okay, so it's, right, does that make sense?
I know that that's why you could use heat, but I don't see how that's information or not.
No, it's conversion of physical information into genetic information. Right, so you had something dead, so you had this dissolved organic matter, which is a physical organization of molecules, right? And then you're breaking those into, you're eating them, right? And converting them into cells and viruses. Right, so your product is, what you do is you watch DOM go down and you watch cells go up. So they're converting one information type into another. Good, does that make sense? Do you see where I'm going there? Yes, no. So this is actually what it looks like if you assume, if you put it on this chart, okay? So right here, this is just the heat that we measured in the calorimeter going on along this side. And then this is my guesstimation of, based on other things I just calculated out of the literature, because we didn't do the metagenomes in this case, right? But this was how many more, how much more genetic information we should have, our metagenome effectively, when we have viruses in total number. And if you do that, you'll actually see something which I don't quite understand why it has to slope down, but in all cases it looks like it slopes down. So that would really suggest that there is actually something on this plot, which is why we have to keep measuring it, Victoria, right? So we have to get a lot of these data points, right? To really make that, regular. Do you guys see that? Okay. And we